Hi, I'm Dr. Devin Chen. I'm a postdoctoral fellow here at the Toronto Zoo, and I work in the Reproductive Sciences Lab. So the species my research focuses on is the Eastern Massasauga rattlesnake. This is a rattlesnake here in Ontario that is threatened with extinction, and so here at the zoo we've started a conservation breeding and reintroduction program. So something interesting about this species is that they are sexually monomorphic, so the males and females look the same from the outside. Also, it takes them around 3 to 5 years to sexually mature, and so in that time it can be difficult to determine the males versus the females. So here at the Toronto Zoo, we've refined a method where we can use molecular genetics to sex individuals using their snake skin sheds, actually. So over here we have an example of one of the sheds. So this is from an adult Eastern Massasauga rattlesnake. And so they shed their skin naturally throughout their life while they grow. And so we can take this from their habitats, their enclosures, and we can extract DNA from it. And so this is a non-invasive method. We don't have to touch the animals, which is great because we don't want to stress them out, and they are a venomous species. So this allows us to non-invasively gain information to help with our management and understanding of the species. So what we do is we extract DNA from the skin sheds. So we will grind down the skin sheds and put them through a spin column to extract and isolate just that DNA, and we are looking specifically at the sex chromosomes. So with humans, sex chromosomes, the males have XY sex chromosomes where the females have XX sex chromosomes. With reptiles, it's a bit different, and with the Eastern Massasaugas, the females actually have a ZW sex chromosome set up, whereas the males are ZZ. And so we can use that difference between the males and females to determine their biological sex by looking at their DNA. So we look for a gene that's specific to that Z chromosome, and we put it through what's called qPCR. So that is what this machine is here. And so basically we put our samples through this machine, and it's going to amplify that DNA, and we can look at differences in the amounts of that resulting amplified DNA, and this will tell us if we have a male or a female snake. Get an output that looks like this, and it is from a software that is connected up to our qPCR results, and this helps us determine whether a snake is male or female. So ADARB in green, that's the name of the gene that is linked to the Z chromosome that I mentioned earlier. In red, we have a gene called MECOM. So this is actually an autosomal gene. So these are genes that are on our somatic cells, and people have 2 copies of the MECOM gene. So since males have the ZZ sex chromosomes, they're going to have 2 copies of the ADARB, and they'll also have 2 copies of the MECOM. So we're going to get this overlap on the graph between these 2 lines because they have the same copies of that gene. Whereas when we look at females, since they're ZW, they only have 1 copy of that ADARB gene, and so there's going to be separation because the females, since they only have 1 copy of ADARB, it's going to be farther away from the red line of the MECOM since there is lower gene expression. So this helps us determine whether we have a male or a female snake. We're also applying this technique to snakes in the wild. So we've teamed up with some of our field partners where they are sending us the skin sheds from neonates or baby Eastern Massasaugas, and we are able to use our technique, the qPCR, and determine the sex ratios of those clutches from the wild. This is really important for different population monitoring techniques such as population viability analyses or PVAs, so that they can more accurately monitor and predict the population so that we can help make these populations sustainable into the future to avoid extinction. We've also used this technique to sex some of our individuals here at the zoo that we have bred and raised that were reintroduced to the wild. This is also important because it helps inform our reintroduction efforts to determine if there are factors such as sex-biased dispersals.